Welcome to the Communication and Cell Signaling Pathways lecture. This PowerPoint is going to be based on how cells communicate with one another and how they send signals to go deep into the cell and cause changes within a cell. We'll talk about the types of changes that you may see and the different types of signals that can be sent. So, what are the types of cell signaling? Well, there are 5 primary types. There's gap junctions, and as we talked about in previous lectures, gap junctions are simply openings like tunnelways between cells, and they allow for cytoplasm to flow back and forth freely, allowing things like enzymes, hormones, signaling molecules to move back and forth as well freely between 2 cells. There's contact-dependent signaling, where one cell has the signaling molecule and the other cell has the receptor. And these cells must come into contact for the molecule to bind to the receptor to cause some sort of intracellular change. Then you have autocrine and paracrine signaling. The term auto, anytime you see that prefix from this point forward in this class, always think of the word self. Autocrine signaling is self-signaling. That is this first cell here. This first cell releases the signaling molecule and binds it to its own self-receptor. So this is autocrine signaling. Paracrine signaling, para means next to. So let's say this cell releases the signaling molecule and that molecule can bind to neighboring cells. That would be paracrine signaling. The next type of signal is a hormone signal. And the key thing with hormone signals is twofold. Number 1 is that you have endocrine cells making the hormone, and that hormone must travel through the blood. That is rule number 1. All hormones must travel through the blood circulation. Rule number 2 is that for that hormone to be able to cause some sort of change, it has to have the proper receptor to bind to. So if you see this cell here doesn't have a receptor, the hormone's going to have no function on it. But the cell with the proper receptor will bind the hormone and then create the response. So not only is it important to make the right hormone, but it's also important to have the correct receptor present. The next type of signal is a type of long-distance signal, and these are called neurotransmitters. Neuro, because they're made from neurons, and transmitters because these guys are not hormones, they're just chemical signals that are released onto receptors on target cells. Now, I made it a point to tell you that these are not hormones because as neurotransmitters, these do not need to travel through the blood. However, there are types of signals called neurohormones. Again, neuro because they're released by neurons. And the word hormone indicates that it is a hormone chemical signal that again must travel through the blood and bind to a proper cell with a proper receptor in order to have a response. The last type of signaling molecule is one that's solely in your immune system. These are called cytokines. All of these are types of cytokines. You do not need to memorize them, but just see the variety of them. Cytokines are specifically signaling molecules of your immune system. You have ILs, which are interleukins, TNFs, which are tumor necrosis factor, and all of these guys are released between immune system cells to communicate between different cell types, like your B cells, like your T cells, like NK cells, which are natural killer cells, and between all your white blood cells. Now, with that being covered, we are now going to go into one of your first big lectures with physiological mechanisms. I will let you know that this, the remainder of this PowerPoint is a huge part of your next exam. These are the mechanisms that we'll be talking about. There are 3 specific ones that we're going to go through the mechanisms for. But all in all, there are 4 different types of cell receptors, but I'm only going to go through these 3. Receptor channels, receptor enzymes, and gproteincoupled receptors. They each have mechanisms, and you must memorize these pathways. Before we can talk about the mechanism of the transporters, we do have to cover one chemical called CAMP, cyclic AMP, and that's right over here. Cyclic AMP is what we call a second messenger, and this is the chemical that actually sends the message downstream inside of the cell. So when we start learning about these transporters and these signaling mechanisms, you'll see that the signaling molecule binds to the receptor outside of the cell and doesn't actually ever get into the cell. It stays outside, bound to the receptor. But what it does is it triggers production of CAMP in the cell from ATP. So although this might not make sense right now, in the second or third mechanism I'm going to teach you, we're going to come back to this. But I do need you to know that CAMP is considered a second messenger that is responsible for sending a downstream signal into the cell. We're also going to cover IP3 and DAG in a separate mechanism, and again, these two are second messengers that send intercellular signals in through the cell. So just to kind of give you an idea of what that CAMP looks like is here's your signaling molecule, which we'll call a ligand. It'll bind to the receptor. When the ligand binds to the receptor, it becomes activated. This receptor will then activate another protein in the membrane. We call this protein adenylyl cyclase. Make sure you know this name. Adenylyl cyclase converts ATP in the cell into CAMP. The job of CAMP is to then go and activate other downstream proteins to create some sort of cellular response. So again, the signaling molecule itself doesn't get into the cell, but it does trigger a cascade of events that produces CAMP. CAMP activates other downstream factors that cause a cellular response. This slide is not in the PowerPoint, so if you want to pause it and draw this out right now, I think that might be helpful. So here is our first signal receptor. We're going to call this the tyrosine kinase receptor. Tyrosine kinase receptor is a type of receptor that spans the entire cell membrane, from the extracellular fluid side to the intracellular fluid side. What you'll see is that you have the ligand, the signaling molecule, you have the receptor, you have part of the protein that's connected to the intracellular portion of the protein, which we call TK, the tyrosine kinase. So when the ligand binds to the receptor, it causes the receptor to shift shape just a little bit, causing almost like a domino effect to shift the shape, moving downstream, eventually activating the tyrosine kinase intracellularly. When you activate tyrosine kinase, its job is to take a protein and an ATP and phosphorylate that protein using that ATP. Now, if you recall from one of the first PowerPoints we went through, you know that ATP is adenosine triphosphate. It has 3 phosphate groups on there. What this tyrosine kinase does is it takes one of the phosphate groups from ATP, plucks it off, and binds it onto the protein, activating this protein now. That is essentially the job of all kinases. So whenever you see this word kinase, this is just a big umbrella term that just means some sort of molecule that phosphorylates something else. So a tyrosine kinase phosphorylates these proteins intracellularly. Kinases will predominantly get their phosphates from ATP because there's 3 phosphates on here. So you pluck a phosphate off, and then you bring it with the protein, and it turns into a phosphorylated protein. One of the byproducts ends up being ADP, adenosine diphosphate. So you went from triphosphate to diphosphate because one of the phosphates is now here bound to the protein. You have now successfully activated a protein in the cell by simply binding this signaling molecule to that receptor. Again, notice that this signaling molecule doesn't actually get into the cell. All it did was bind to the receptor, cause some sort of structural change to activate the tyrosine kinase, And once this was activated, it acted as a kinase would, and it phosphorylated the protein to activate it. The next type of receptor that we're going to talk about is called a gprotein-coupled receptor, or a GPCR. These receptors, again, span the entire cell membrane. They have a cytoplasmic side and they have an extracellular side. These mechanisms here have a lot of players in them, so I'm going to go through them very slowly. I'm going to be very repetitive so that you guys have enough time to maybe take some notes or just hear it a second time to help you remember it. So this is where the mechanism starts. This is called the GPCR adenylyl cyclase, a G protein couple receptor that uses adenylyl cyclase for transmitting a signal. Here's your ligand, aka your signaling molecule. In beige, you have your G protein coupled receptor, so it's the receptor. And what you'll notice is that this cytoplasmic end is bound to a G protein, which is made up of 3 different pieces that are together. So ligand, the receptor, and the G protein. Before we talk about the actual mechanism, G proteins have the ability to break off one of their pieces and send them down the membrane. And you're going to see that happen in this mechanism. So step 1, the ligand binds to the GPCR. The ligand binding to the GPCR causes a domino effect of structural changes along this protein. Because the GPCR changed shape, the G protein is also going to change shape. Anytime you shift the structure of a protein, it's going to cause a downstream change as well, and it'll change any protein that's bound to it. So the ligand binds to the receptor changes shape, which causes, like a domino effect, the G protein to change shape. When this G protein changes shape, it activates. And what happens is one of the pieces of the G protein will travel down the membrane, and it'll bind to this other protein here, called adenylyl cyclase. If you recall, adenylyl cyclase is the protein that converts ATP into cAMP. So we're going to have that as our next step. So again, ligand binds to the GPCR, activates it, which causes activation of the G protein. G protein breaks off one piece to travel down the membrane, bind to the adenylyl cyclase. Because you bound a piece of the G protein to the adenylyl cyclase, it activated adenylyl cyclase to turn ATP into cAMP. And like we said, the cAMP is the second messenger, and it has the ability to then activate other things downstream. cAMP's job is going to be to activate protein kinase A. Now the word kinase is in there. What do you know that term means? What do kinases do? Well, kinases phosphorylate other molecules. So each of these kinases has the ability to exponentially activate other proteins downstream. Those proteins activate other proteins downstream as well. So what you have is an exponential cell response. Because of one ligand binding, you were able to activate exponentially and create a huge cellular response in the cell. Please be sure you know the names of all of these enzymes that ATP converts to cAMP. cAMP activates protein kinase A, which is also abbreviated pKa. pKa will phosphorylate proteins, activating them to then activate other things downstream to cause some sort of cell response. The next type of GPCR we have is called the phospholipase C signal transduction. Now, the beginning of this mechanism is identical to the last one. The ligand binds to the G protein coupled receptor, shifting its shape, which causes a downstream shift and activation of the G protein. Now, from the last mechanism, we learned that the G protein will then activate adenylyl cyclase. But in this one, this is where it changes. Instead of the G protein activating adenyl cyclase, it activates a different enzyme called phospholipase C, PLC. Down here in the legend is the names of everything that I'm going to use abbreviations for. So once G protein activates phospholipase C, this membrane enzyme is going to take the membrane phospholipids and break them down into IP3 and DAG. Now, where a lot of students get confused is, hey, where did this come from? Well, if you recall, all cell membranes are lipid bilayers. Lipid bilayers are made out of phospholipids. So PLC is essentially literally taking the phospholipids from the lipid bilayer and it's breaking them down into two different other molecules, IP3 and DAG. Because this membrane is mostly hydrophobic, we can also predict that DAG is hydrophobic because it stayed in the membrane with that hydrophobic region. The cytoplasm is mostly hydrophilic, so you can probably guess that IP3 is also hydrophilic. As you know from chemistry, like dissolves like. Things that have similar characteristics like to stay with each other. So again, the ligand binds to the receptor, which activates the G protein. The G protein activates the PLC. The PLC is an enzyme that takes a membrane phospholipid and breaks it down into DAG, which stays in the membrane, and IP3, which goes into the cytoplasm. Now, we're going to go through what each of their functions is, starting with DAG. DAG will then go downstream further and activate PKC, protein kinase C. Again, the word kinase is in here. What do we know that all kinases do? Well, they phosphorylate things. Protein kinase C will phosphorylate a protein. This PI, that means it's a phosphorylation group. It means inorganic phosphate. So you add the phosphate group to the protein through PKC, and you get an activated protein here. Again, just like the previous mechanism, this protein can activate other things downstream and create some sort of cell response. So that's the DAG pathway. The IP3 pathway is a bit more simple. In the cytoplasm, IP3's job is to go to the ER, the endoplasmic reticulum. One of the main jobs of the ER is to store calcium. When IP3 binds to the ER, it causes opening of calcium channels. Now, because calcium is in such high concentration in the ER and in such low concentration outside in the cytoplasm, that causes calcium to want to diffuse out. That is the definition of diffusion, you must know. Diffusion is the movement of solutes from areas of high concentration towards areas of lower concentration. So the minute IP3 binds to the ER, it's able to open up the calcium channels in the ER, allowing calcium to diffuse out. Now, just like a phosphorylated protein, calcium also can act as an intracellular signaling molecule, activating other downstream responses. That is this entire mechanism here. Make sure you go over this several times. Try to draw it out on your own with just this list here, and try to either teach it to somebody else, which is honestly the best way to learn this, or record yourself going over it, because once you can go over it on your own without looking at this picture, at that point, I think that you have the material down. Now, the next type is going to be a receptor channel mediated mechanism. And again, it's receptor channel mediated, so there's a receptor on a channel. Ion channels are little tunnels that are found in the membrane of cells. Channels have the ability to be opened or closed. You see this kind of ball and chain looking guy? It's open right now. but this ball can easily come and block that channel, and it would be a closed channel. So what dictates whether a channel is open or closed? Well, binding of signaling molecules. So receptor channels can open or close in response to binding of signaling molecules to the receptor site. Now, it can either bind directly to the receptor site, or there's a second way. Signaling molecules can bind to the gproteincoupled receptor, which, as we know, activates a G protein. As we know, this activated G protein can move downstream and bind to something else. That something else could directly be an ion channel. When G proteins bind to ion channels, it can cause them to open or to close. So two ways to actually open and close ion channels. One way is to bind the signaling molecule directly to the ion channel. The other way is to bind it to the receptor, to activate the G protein. Then the G protein interacts with the ion channel in order to open or close it. Now, one rule about ion channels is that they are specific to the ion that they want to transport. So calcium channels, they only transport calcium. Sodium channels only transport sodium. So they're very specific in terms of what they move downstream. The second thing about ion channels is that they work based on diffusion. They move ions down their concentration gradient. So let's say there was very high sodium on the outside and low sodium on the inside. When you open a sodium channel, which direction do you think sodium is going to flow based on diffusion if it's high concentration on the outside and low concentration on the inside? Sodium is going to definitely want to flow into the cell because per diffusion, solutes always want to go from an area of high concentration down their gradient to an area of lower concentration. So with that being said, once you can signal the opening of ion channels, it's going to change the permeability to ions like sodium, potassium, and chloride. And by no means are these the only ions that move through channels. They're just the three main ones that I mentioned. And once these ions move, I want you to notice they have charges to them, positive and negative. I'm going to write this out, and I'd like you to write this as well. The resting voltage of a cell is negative 60 millivolts. This is where a cell likes to be at rest, okay? All cells in your body are at a nice, even negative 60 millivolts. That is the inner voltage compared to the outer voltage. So it is safe to say the inside of your cell is 60 millivolts less than the outside of your cell. What do you think is going to happen if you open a sodium ion channel and you allow sodium that has a positive charge to start moving? As you know from one of our first lectures, there is a lot of sodium on the outside of a cell, but there's very little sodium on the inside of the cell. So naturally, per diffusion, when you open a sodium channel, sodium is going to want to move into the cell. If sodium has a positive charge and it's moving into the cell, what is the overall voltage of the inside of the cell going to do? Is this voltage going to go up closer to 0, or is it going to get more negative, let's say closer to negative 100, if you have positively charged sodium moving into the cell? Well, if you said that this voltage is going to rise up towards 0, you would be correct. When you bring a positive charge into the cell, you're going to turn this negative 60 into a negative 50, into a negative 40, negative 30, and so on and so forth. It all depends on how much sodium you allow into the cell. So, changes in voltage create electrical signals. Go ahead and write that down for me. Changes in voltage create electrical signals. And why is that significant? Because Electrical signals have the ability to activate or inactivate voltage-sensitive proteins. There are proteins that can become active because you made the voltage higher. There are proteins that can become inactive because you made the voltage higher. They're very interesting proteins that are controlled by voltage, and we'll talk just about, I think, one or two of them this entire class. So this is the point where we go to the next slide, and I explain this part more in depth. So let's say you allowed sodium into the cell. The voltage of the cell rose up, and that created an electrical signal. That electricity shoots through your cell, and it can activate other proteins. And as you know, channels are types of proteins. This channel here, it's literally called a voltage-gated calcium channel. That's the entire name for it. I know it's long, but it's okay because it actually tells you what the function is. It is a calcium channel, which means it allows calcium in and out based on the voltage. This channel opens up only when voltage shoots through the cell, and it is a rise of voltage that will open up this channel. So when the electrical signal shoots through this cell, that electrical signal will eventually reach a voltage-gated calcium channel. When it reaches the voltage-gated calcium channel, it opens it. Now again, from one of the previous PowerPoints, I think it was the first one, calcium is always found in high concentration outside of your cell and low concentration inside of the cell. Make sure you memorize that fact. Calcium is high concentration outside of a cell and in low concentration inside of the cell in the cytoplasm. So what do you think is going to happen when you open up a voltage-gated calcium channel? Will calcium want to enter the cell, or will calcium from in the cell want to leave the cell? Well, if you said calcium is going to enter the cell, you would be correct. The voltage-gated channels open, allowing calcium to enter into the cell, and this calcium, as we know, acts as an intracellular signal. The second thing that happens from this voltage change is that this voltage can also activate those calcium channels on the ER. Remember the ER, it is a huge storage of calcium. So when that voltage shoots through the cell, it not only goes through the membrane, but it also goes internally. The voltage hits the ER, it opens up voltage-gated calcium channels in the ER, and that causes release of calcium from ER out into the cytoplasm. Overall, what you're doing is you're increasing the amount of calcium in the cell, which causes an intracellular signal. This calcium has the ability to bind directly to proteins, and it can cause changes in the protein activity. It can cause a cell to want to do exocytosis, or it can cause a cell to move like it does in muscle cells. All of these transporters that I just covered right now, they all have a little bit of overlap. And it's important to be able to draw these guys out on plain white paper and make sure you understand the step-by-step. If you are confused on any of these channels, this is a wonderful time to ask me questions during our next Zoom meeting, or to email me, or to schedule a Zoom meeting with me personally. So hopefully that wasn't too brutal just yet, but that is examples of mechanisms and how they will be taught as a step-by-step. So one thing happens, causes another thing to happen downstream, and that will cause several other things to happen downstream. Now, we have ways to control these signals. Yes, you made a ligand. Yes, it bound to a receptor. But what else is there? Well, there's specificity, there's competition, there's antagonists, and there's different types of receptors. So we're just going to really touch briefly on these things. The first thing I want to cover, which is a really, really interesting slide here, is the type of target receptor. When you make a ligand, that ligand binds to a receptor. But it's not just about what ligand you make. Like this example here, you made a ligand and it's epinephrine. Now, epinephrine has two different receptors it can bind to. It has alpha receptors and it has beta receptors. One place out of many that alpha and beta receptors are found is going to be on blood vessels. There's alpha receptors on blood vessels and there's beta receptors on blood vessels. They're found in different systems. For example, there's alpha receptors in the intestinal blood vessels and there's beta receptors on the skeletal muscle blood vessels. So before we move on, let me kind of tell you what epinephrine is. Epinephrine is adrenaline. Let's say you get scared and your body releases epinephrine. That epinephrine is there to help increase your heart rate. It's there to help you breathe faster. It's there to dilate your pupils. All the things you want to happen to your body during a scary situation, epinephrine does it. At the same time, epinephrine is also able to downregulate organ systems that you don't need when you're scared. When you're scared, you don't need your urinary system. You don't need your reproductive system, nor do you need your digestive system. So epinephrine's job is twofold. Number one, activate all the organ systems you need. Activate the brain. Activate the skeletal muscle to run away from something scary. And it also is there to downregulate the systems you don't need, so we don't have to waste energy on them. And with that being said, let's look at these examples again. Epinephrine can either bind to alpha receptors that are found in the intestine or beta receptors that are found in your skeletal muscle. Again, which one of these places is going to be a place you don't need during a panicking response? Well, if I'm panicking, I want to run away from a situation. This is something I do need. If I'm panicking, I'm not thinking about a peanut butter and jelly sandwich. So intestinal and digestive tract is something I do not need. So what epinephrine does is it goes and binds to both receptors at the same time. Why? When epinephrine binds to alpha receptors on the intestinal blood vessels, it causes vasoconstriction. When you close up the blood vessel, less blood gets to that organ, which is fine because you don't really need it at that moment. It detours the blood from here to places where you do need it, like your skeletal muscle. So when epinephrine binds to beta receptors on skeletal muscle, it causes the blood vessel on the skeletal muscle to dilate to allow more blood to go to that region. So it's always a matter of not just the type of signaling molecule you release, but also the type of receptor it binds to. Then you got agonists versus antagonists. I like to think of these guys as our pharmaceutical players. So a lot of medications, they work to either upregulate a system and act similarly as a naturally made chemical, or to downregulate a system to block a chemical from actually binding. So let's say you have a chemical in your body like testosterone. Testosterone binds to an androgen receptor to cause whatever response it has to cause. Men who are low in testosterone sometimes take a testosterone supplement. That is called an agonist. It is a different signaling molecule that can still bind to the same receptor to have the same response. However, some men make way too much testosterone, and overproduction of testosterone actually gets converted into estrogen, and it causes men to lactate and develop breast tissue. To stop that, pharmaceutical companies have made antagonists to receptors of testosterone. These medications bind to the testosterone binding site and block it, so that testosterone itself cannot bind to the receptor and therefore does not cause any downstream responses. So two key terms here, agonist and antagonist. So I don't need you to memorize these by any means. This is just for you to understand how different pathways work and how they signal in just your endocrine system, in just your nervous system, and in the system that combines nervous and endocrine systems. These last two slides, what I need you to know from them is the major differences. And what I'm going to do is I'm going to take my pen and I'm going to star the main characteristics that I'd like you guys to know. So we're looking at the differences between neural and endocrine reflexes. So just nervous system and just the hormone system. In the nervous system, the signal is electrical. In the endocrine system, it's a hormone, so it's chemical. The nervous system response is very fast, and the response time for the endocrine system is much slower. It takes a lot more time for hormones to actually go through your blood and bind to a receptor. The duration of the nervous system reaction is very short. You send a response and then it's done. But for the endocrine system, once a hormone binds, that hormone is going to be bound for a good amount of time. These are the three from each side that I need you to know the differences of. Now, the next slide shows you something similar, but in the middle, it shows you what the neuroendocrine system does. This is a system that uses neurons, but instead of sending electrical signals, they send hormone signals. So just be sure to go over this slide and make sure you understand the differences between neural, endocrine, and neuroendocrine. With that, that is the end of the cell signaling PowerPoint. Please be sure to go over this at least twice to make sure you understand all those mechanisms because they're really going to help you later on in this class.